Welcome to my chemistry and captivity video project. How creatine actually works. This is a breakdown of the specific chemistry behind the supplement creatine. I enjoyed this project a lot, so let's get into it. So in this video, we're going to go over what creatine is chemically, how your body creates creatine and what it's made out of, how creatine enters your muscles from the sites where it's created, what creatine kinase and phosphocreatine are, creatine phosphocreatine shuttle, which is like the pathway that the creatine goes through to be used, what changes to how your body functions when you have extra creatine in your system via supplementation, and the long-term fate of creatine, which is basically just how it's cycled through and how it's disposed of when your body is done using it. So what is creatine? Creatine is a organic compound, which means it has a carbon-hydrogen backbone. It is created from these three amino acids right here: arginine, glycine, and methionine. It's built out of these via a process called SAM. 95% of your creatine is stored in skeletal muscle, mostly as phosphocreatine. The other 5% is stored in your brain and your heart in order to help regulate ATP use. So, how is creatine made and where does it come from? It's made with those three amino acids: glycine, arginine, and methionine down here. The glycine is going to be the backbone of this molecule, so it is what everything else is going to combine onto in order to develop creatine. This arginine here is going to give this guanidino group to the glycine right here to create guanidino acetate, and then the methionine is going to add a methyl group to create creatine. This takes place in the kidney and liver via SAM, which we're about to go over. So the first step to creatine synthesis starts in the kidney, where this enzyme, AGAT, helps combine the arginine and the glycine by taking this guanidino group off of the arginine and putting it onto the glycine, which outputs guanidinoacetic acid. Without that guanidino group, this arginine now becomes orthonine, which is just another byproduct of this reaction, which is used for other stuff in the body. But this molecule right here, this guanidinoacetic acid, often referred to as GAA, is then sent over to the liver, where there will be another process that finishes the creatine synthesis. So the second step to creatine synthesis occurs in the liver, where this enzyme called GAMT helps regulate the reaction between GAA, guanidinoacetic acid, and SAM, which is basically just the methionine. This is going to allow that methionine to donate a methyl group to the guanidinoacetic acid, which creates creatine, and then SAH, which is just the methionine without the methyl group. In the end, this outputs creatine. So after your creatine is created in the liver, how does it get into your muscle cells to be used? So after it's in the liver, it gets expelled into your bloodstream, where it then has to get into your muscle cells. But unfortunately, your muscle cells have this membrane around them, this phospholipid membrane around them, that holds the cell together, but also prevents stuff from just coming into the cell when it wants to. So you can't just have the creatine flow straight from your bloodstream into the cell. So what your cell has, are these little transporters on the sides of the membrane, which are kind of attached to the membrane, and they're basically like a little tunnel that the creatine can take to get in, and it can only get creatine in because of how the chemical structure of it is set up, which allows your cells to receive the creatine while also keeping out things that they don't need. So after creatine is in your cell, this enzyme called creatine kinase catalyzes this reaction where the creatine combines with an ATP, which is adenosine triphosphate, to take one of those phosphate groups off and add it onto the creatine. This allows the creatine to become phosphocreatine, which has it storing a lot of energy. So this stores one phosphate group off of that ATP on each creatine, and that allows it to store this energy, which can then be re-put into the creatine kinase and come out back as creatine, but also now your cell can reuse this phosphate group later when it wants to. It doesn't have to, create more high energy phosphate groups on the fly. It can hold it on this phosphocreatine and then take it off later when it needs it. And that's really at the core. That's what creatine does. Is it just stores energy for your cells so that your cells have quick energy to get to when they need to. So now we're going to get into the creatine phosphocreatine shuttle. So this is basically just the process that your cells go through to take that ATP, put it on the creatine with that creatine kinase, turning it into phosphocreatine, and then used it in the muscles, and then going back to the cell to be reused again. So now we're on shuttle step number one, which is the mitochondrial side of things. This is where your cells go through a really important system of reactions called oxidative phosphorylation, which is really just how your mitochondria uses glucose and turns it into ATP, this high energy molecule that can be used to do cellular functions. Um, Now, what happens is your cells create, your mitochondria creates this ATP, which then combines with the creatine via mitochondrial creatine kinase, which is just creatine kinase that is located near the mitochondrial membranes, waiting to combine the ATP and the creatine into phosphocreatine and ADP. So what that does means is that it just takes those phosphate groups off of the ATP and puts it onto the creatine, which is allowing it to start storing energy. So after the mitochondrial creatine kinase turns that creatine into phosphocreatine, the phosphocreatine is going to leave the mitochondria into the cytosol in order to go find where it needs to go. Now, what happens is because there's areas in the cell that are burning ATP, they're turning from ATP into ADP. Now, that's going to cause there to be a higher concentration of ADP versus ATP in that area, which is going to cause the phosphocreatine to diffuse into that area, pulling itself in via the different gradients, where there's another creatine kinase in that area. This creatine kinase is called the muscle cytosolic creatine kinase, and this is where the phosphocreatine is going to turn back into creatine via that muscle cytosolic creatine kinase, pulling that phosphate group off of the phosphocreatine and using it in whatever function the cell needs it for. Then the creatine is going to get sent back to the mitochondria via the same diffusion tactic as earlier, and the process will start over again. So, in summary, your kidneys help to combine arginine and glycine into guanidinoacetate, which then gets turned in to creatine in your liver via GAMT combining the guanidinoacetate and SAM. Then after that, it's excreted into your blood from your liver, which is then transported to your muscle via SLC6A8. And then phosphocreatine creatine shuttle then buffers ATP and helps regulate energy during rapid high intensity exercise.